we will explain about serum therapy.
First, what is serum therapy? In Japanese, serum therapy is translated as 血清療法. Then, what is serum? Human blood is mainly composed of two parts, plasma and blood cells, such as red and white blood cells. Plasma is a yellowish fluid consisting of about 19% water and dissolved substances, including antibodies like globulins. When fibrin, the component responsible for blood clotting, is removed from plasma, the remaining fluid is called serum.
Let's look at the background behind the emergence of serum therapy. Kitasato Shibasaburo is best known for one of the greatest accomplishments of his career. He was the first in the world to successfully obtain a pure culture of tetanus bacterium. Tetanus bacteria exist naturally in soil and the feces of animals. What is the tetanus bacterium? At the time, it was widely believed that tetanus bacteria could not be cultured because it was not yet known that they were anaerobic bacteria. Since they die in the presence of oxygen, the prevailing belief was that pure culture was impossible. However, Kitasato recognized their anaerobic nature. In 1889, he succeeded in culturing the bacteria, and the following year, he used a large amount of toxin obtained from the culture to develop what we now call serum therapy.
How does serum therapy work? Serum therapy involves administering serum that contains antibodies using the antigen-antibody reaction to treat disease. An antigen-antibody reaction occurs when an antigen, something foreign to the body, binds specifically to an antibody made against it. This reaction neutralizes toxins or helps the immune system remove harmful substances.
For example, if a horse is repeatedly injected with a toxin, the horse produces antibodies against it. The antibody-rich serum collected from the horse is called immune serum. When this immune serum is injected into a patient, the antibodies bind to the antigen in the patient's body, helping to improve the symptoms.
How is this different from vaccines, such as influenza vaccine? A vaccine contains a weakened or inactivated form of pathogen or its toxin. It stimulates the body to produce its own antibodies. Serum therapy, by contrast, uses antibodies produced by another animal.
Is serum therapy still used today? Modern medicine has produced far more effective treatments. Therefore, serum therapy is no longer commonly used. However, serum therapy is still valuable in certain situations, especially in an emergency. For example, if snake bite emergencies, it takes time for the patient's immune system to produce antibodies. To prevent severe symptoms, immune serum containing anti-venom antibodies produced in animals is administered. The patient's own immune response, combined with the injected antibodies, fights the toxin more effectively. I will explain strengths and weaknesses of serum therapy. One major advantage is that it is effective without having to wait for the body to produce antibodies. On the other hand, because the body does not produce many of its own antibodies, the effect is only temporary and allergic reactions to animal-derived serum can occur.